All right. I'm hoping that this works because I'm just gonna keep going here. I should be recording. Oh, I should have hit pause record. Dadgummit. Now I'm gonna have two files. Ugh, this digital nonsense. I've gotta catch up to the digital age here. Actually, not really. I'm pretty much there. I just didn't notice that. Anyway, that brings us really to acids and bases. This idea of hydrogens and hydroxyl ions, that is kind of what makes up acids and bases. Acids will dissociate in water to produce this positive hydrogen ion and an anion of some sort. They become this proton donor. We increase the concentration of that free hydrogen ion, and as we as we see more of those free H pluses, those free hydrogen ions, we're going to start to see them dissociate more of those hydrogens off of whatever substance it's encountering, which is why acids melt your face. It's because the acids will quite literally break apart the molecules trying to get at all of the hydrogens that are present in your tissue. And so stronger acids, of course, are going to dissociate more and more of those hydrogen ions, and weaker acids not so much. But these different acids are, they're going to control different things. So, your carbonic acid, as carbonic acid in the blood increases, that will actually trigger your body to breathe deeper and more quickly, because it's trying to get rid of that carbonic acid eventually. I mean, carbonic acid, it is kind of a weak acid, but it's going to hurt you eventually. And kind of the same thing with your stomach acid. The high acid and that dissociation of hydrogen ions is going to activate certain enzymes that will help to digest your food in your stomach. Talk about that later on in 202. So anyway, so we're going to have this base that will accept a hydrogen ion when added to solution. It's a proton acceptor, and it's going to decrease the concentration of your free hydrogen ions, which is why, for example, bicarbonate in the blood, baking soda, essentially. So we take the baking soda, we take the vinegar. We've all done this before, vinegar and baking soda. It foams up. It's going to react because the bicarbonate in the baking powder or soda is going to, it's uptaking all of those free hydrogen ions in the vinegar. And so if you were to take the baking soda and the vinegar, you mix them together, and now all of a sudden you've got a completely different substance. You've got a lot more water than you do acid. Now, the stronger your base is, the more absorbing those hydrogen ions we're going to see. And we're going to see a little bit less absorption, of course, with the weaker base. But this is also partly why bases can be just as problematic as an acid. Bases can actually take all of those hydrogen ions that maybe your body has hooked up to your skin. So some of you guys know when you pour straight bleach on your hands and you rub your hands, it's kind of slippery. That's partly because the bleach is such a strong base that it's literally melting your skin off. It's taking up all of those hydrogen ions and breaking apart the molecules that make up your skin. So they do very, your bases and your acids can both be caustic, but they're just working in different ways. So let's see here, neutralization. So this is where pH comes in, right? And this is an explanation of what the pH meter, what the pH, what it means. Essentially, it's percent hydrogen is what pH means. And it's an inverse relationship between like a logarithm. So now, you know, we're going from chemistry into math, and like, ew, I'm really not a math guy. So I don't really care if you know this or not. It's interesting, and I kind of understand it. You guys can read it on your own, but I don't really care if you know it or not. It's not going to affect your grade if you know what pH, you know, exactly is. But what you do need to know about the pH is the pH scale. You have to be able to interpret the pH scale. So solutions with an equal concentration of hydrogen ions, and hydroxide ions are going to be neutral, okay? They're balanced out. This is pH 7, okay? pH of 7 is going to be neutral pH. It's not basic, it's not acidic, it's just neutral. It's water, all right? Solutions that have a little bit more hydrogen ion is going to be a little bit acidic, and they're going to have a pH level of lower than 7. So as we go lower than 7 on the pH scale, we see that acidity level going up. Now this relationship is a little bit confusing, so you guys are going to need to remember this. As the pH level goes down, the acidity level goes up, okay? And then with your bases, a solution that has a pH level of higher than 7 is going to be a stronger base. And so as we move up the pH scale past 7, the baseness, we call it alkalinity, actually. The alkalinity goes up as well. And so here it is. Right here, there's neutral. Right there, pure water is neutral. And as we go up, we're going to have, things are going to get more basic or more alkaline, right? My little sister, I was going to make a joke about my little sister being super basic, but maybe I won't. So anyway, so as we go up the pH scale, I don't know why this pH scale is irritating. It's upside down. When I was doing this, going up, I didn't realize that they have it upside down. So as we go up the pH scale to 8, to 9, to 10, 11, 12, things are going to get more and more alkaline, more and more basic. And so seawater is at an 8. An antacid, so a Tums, is going to be 10.5. Ammonia gets even more. Bleach is really basic, because keep in mind that things are increasing by a multiple of 10 or 100. Yeah, tenfold. Yeah, so pH of 6 has 10 times greater concentration of hydrogen ions than water. So every time, so 7, we're going to go to 6. That's 10 times more acidic than water is. And then here, going from 6 to 5, that's even more. That's 10 times more than 6. So 10 times 10 is 100. So, you know, that's going to be 100 times more acidic than water. And then again, we go to 4, 10 times 100. It's always 10 times more, right? And so we get all the way down to HCl, hydrochloric acid. Hydrochloric acid is what you have in your stomach, and it's really, really acidic. And then over here, sodium hydroxide is going to be really, really basic. Household bleach is actually pretty basic too. So that's the pH scale, and you're going to need to know the pH scale. That is one that you have to understand. Neutralization, that's when your acidic or basic solution goes back down to neutral. And a buffer is going to kind of go both ways. So for example, you've got a lot of buffers in your blood. You've got carbonic acid and bicarbonate in your blood. and that's going to help maintain your blood pH. And so carbonic acid is going to take up the extra hydrogen ions from an excess acid, or it's going to donate the hydrogen ions to neutralize the base. But it can kind of do both. It can go either way to sort of keep things at that nice little balanced baseline, like 7.35 in your blood. Mixtures. Mixtures are going to be combining two or more substances. Substances that are mixed, they're not chemically changed, so I can mix up my Kool-Aid and the water is still there, the sugar is still there, the Kool-Aid powder is still there. It's not a different chemical, it's all the same. At this point, it's just mixed up. And we could possibly separate that. So for example, salt water, if I were to mix up some salt water, I could evaporate the water and be left with just the salt. That's a mixture. We have three categories: a suspension, a colloid, and a solution. A suspension is going to be where you have solid particles that are sort of suspended in the aqueous fluid. Colloid is going to be gelatin. They don't settle really, but they can scatter light. Whereas suspension, all of those solids can actually settle out. And then a solution is where everything just kind of dissolves in the aqueous matrix. And so like soda water, there's not any change in color, right? So I can drink my Sprite, and Sprite is the same color as water because everything is in solution. And then the emulsion. I've talked a little bit about the emulsion. So let's see here. Concentrations is really, it's an expression of mass over volume. So it's however much solute you have per volume of solution, right? And so that's a concentration. So a 20% concentration of salt water would be, I've got 20 particles of salt to 100 for every 80 particles of water, because 20 plus 80, if I've got 20% salt, 80% water, that means I've got 20 particles of salt and 80 particles of water, or I've got 20 grams of salt per every 80 grams of water. So molarity and molality, you guys are going to need to know this for nursing, but you don't need to know it in my class, so we're skipping it. Moles, you don't need to know about any of that stuff. Now, macromolecules, this is where things get a little bit more important. You need to understand your macromolecules. There's 4 of them. Generally, we're going to see a carbon skeleton with a functional group. So let's take a look. Nope, I need to go backwards. Functional groups, most functional groups are polar. Some act like acids, some act like bases. Brings us to polymers. A polymer is going to be a group of repeating subunits, right? So for example, a starch. A starch is going to be, one molecule of starch is going to have a whole bunch of glucose molecules chained up next to each other. So they're all bonded, and they're all bonded covalently. So we have a starch molecule, but it's made up of these smaller molecules of glucose. So the smaller molecule is going to be the monomer that is then bound together in a chain to form the polymer. Usually when we talk about polymers, it's in the context of like plastic, but polymer is a chemical term that we use to indicate that it is a chain of smaller molecules, monomers. So here we have right there, we've got a monomer, and we use a dehydration synthesis in which we're going to take the HO from that monomer and the H right there, we're going to put those together, and create a water right there, right? So we're going to pull a water out of the two monomers, and then that extra space right there is going to bind with that extra space right there to form the polymer, or the chain of molecules that is bound together. This particular polymer is going to be a dimer because there are two. There's two monomers together. But that's essentially what we're doing is we're taking the water molecule out to generate some room so that we can bind these two sites together to form the chain. That is a dehydration synthesis. We're going to dehydrate it. We're going to take the water out of it in order to form something, to synthesize something. And then we've got this other one, which is hydrolysis, which is, it's just the opposite. We're doing the exact opposite thing. We're going to take the water, we're going to put the hydrogens back on those two spaces in order to break that stuff apart. And so we talk a little bit about dehydration and hydrolysis in particularly nutrition. So it's kind of important, but you probably won't have a whole lot more of it in this particular class. So that brings us to the 4 classes of these macromolecules. The first one, lipids. Lipids are fats and oils, okay? When you see the word lipid, your mind should immediately go to fats and oils. Bacon grease, olive oil, Crisco, butter. I'm trying to think of anything else. All of those things, those are lipids, and they're not soluble in water because they are non-polar. They're always non-polar. They do not stick to water. We have 4 classes of lipids, and so there's some word association that you guys need to do with these terms, right? Lipids are fats and oils, and you've got triglycerides, phospholipids, steroids, and eicosanoids. Eicosanoids, I don't know how to pronounce that because I've never used it. You guys won't either. We'll talk about that in a minute. So triglycerides are really great for long-term energy storage. So we form these from a glycerol and 3 fatty acid chains. The fatty acid chains can vary in number and length, and the number of double bonds. So a saturated lipid, a saturated triglyceride, is gonna be completely straight. There's no double bonds. I've got some pictures coming up, as you can see. There's no double bonds on those fatty acid chains, and so they stack up actually very neatly. Saturated fats are almost always a solid at room temperature. So like bacon grease, butter, generally animal fats are going to be saturated with hydrogens, and they stack up and form a nice little solid, semi-solid structure. Unsaturated fats are going to have 1 or more double bonds in those fatty acid chains, and because of that double bond, it causes the fatty acid chain to kink out a little bit, which prevents it from stacking up nicely. So the unsaturated lipids or unsaturated fats are going to be your fats like oil. They're liquid at room temperature. And then polyunsaturated are where we see 2 or more double bonds. So oftentimes, we read on our ingredients list of Pringles, monounsaturated fat, polyunsaturated fat. Unsaturated just means that there's going to be a double bond and that it's liquid at room temperature. So, and yeah, adipose tissue, our fat tissue, is going to store those triglycerides, man. Lipogenesis is the formation of triglycerides when conditions of excess nutrients exist. And lipolysis is going to be the breakdown of the triglycerides when nutrients are needed. So there they are. So really quick, I want you guys to look at these, and I want you to tell me which one is the unsaturated fat and which one is the saturated. Oh, dadgummit, they're both unsaturated. So never mind. So what I want you to notice here, and it's not like you could say anything anyway, because this is a recording. we've got a double bond here, we've got a double bond right there. So those double bonds are going to mean that this is an unsaturated fat and will be liquid at room temperature. So then we get to phospholipids. This is an amphipathic molecule. We already know about it. It's got a hydrophobic tail and a hydrophilic head. Steroids are a little bit different. They don't really have the fatty acid chains like you see in a glyceride. Steroids are composed of hydrocarbons, but they're arranged in rings. And so in a lot of cases, we're going to see like cholesterols. Cholesterols are steroids, steroid hormones like testosterone and estrogen, and then bile salts. And bile salts are kind of important as steroids. And then these eicosanoids, eicosanoids, I don't know what they are. It's a modified 20-carbon fatty acid. Essentially, we're going to have, really, the only ones that you guys might even recognize are going to be prostaglandins and prostacyclins. We don't really talk about these guys in a whole lot of detail. So, yep, animal fats are saturated, vegetable fats are unsaturated, and we can see the partial hydrogenation of unsaturated fat, and that leads to trans fats, of course, which we think increase the risk of heart attack and stroke. So that's like Crisco, vegetable shortening. Yeah. Then we get to carbohydrates, the second class of macromolecules. Carbohydrates are sugars. It doesn't matter what kind of sugar, what form it is in. It is always broken down into sugars. So that bread that you eat, doesn't matter. Rye bread's great. It's nice, it's savory. It's got a real robust kind of flavor. Not sweet at all, but it gets broken down into sugars because it's made up of sugars. It's just starch. We've got three really. I usually just talk about mono and polysaccharides. Monosaccharide is going to be the monomer of a sugar, right? Disaccharide would be two sugars, and then polysaccharides are going to be formed from many monosaccharides. Let's take a look here. Glucose is a monosaccharide. It's a 6-carbon carbohydrate, and it's really common, really common monosaccharide, and very commonly used in the body. And then we've also got glycogen. Glycogen is kind of cool because glycogen is a polysaccharide. It's a polymer. It's going to have a whole bunch of glucose molecules bound together. And it's really great for storage. We can take all of those sugars and we can store them on your liver in the form of glycogen. And, you know, as you need a little bit of extra glucose in your blood, your liver can hydrolyze that. It can perform that hydrolysis reaction to separate the glucoses out. So yeah, there's our glucose, there's our glycogen. Monosaccharide, polysaccharide. Monomer, polymer. Because we've got all the different molecules hooked up to one single molecule. So we also have a couple other ones. I mean, there's some hexose monosaccharides, like your glucose isomers. There's the 5-carbon monosaccharides, which are pentose sugars, ribose and deoxyribose, as well as fructose. And then disaccharides are just a couple of simple sugars bonded together, like sucrose, lactose, and maltose. Those are the most common ones, and certainly we eat a lot of sucrose and drink a lot of lactose. So polysaccharides, glycogen is going to be the most common one found in animals. Starch and cellulose are found in plants. So plant starch is a pretty major nutritional source, of course. That's where we get most of our carbs. Cellulose, however, is a little different. It's non-digestible. We don't have the machinery or the symbiotic relationships to digest it. And so it becomes a really great source of fiber. It just goes right through, sails right through the body, scraping things out. It's good for you. So here's a couple more, yeah. Oh, just pictures, sucrose, lactose, maltose. Note that they're very similar. Lactose and maltose are almost identical, except that, you know, the bond is slightly different. So these are isomers of one another. Then we get to nucleic acids, the third one. Nucleic acids are nice and simple. They store and transfer genetic information, pretty much. That's pretty much what they're used for. You've got two classes, deoxyribonucleic acids and ribonucleic acids. Both of them are storing genetic information. In this particular molecule, you're going to see these three components: a sugar, a phosphate, and a nitrogenous base. We don't need to really go into that in a whole lot more detail. It's got a sugar-based phosphate. Anyway, we've got 5 types of nitrogenous bases: pyrimidines and purines. Again, you don't really need to know a whole lot about this. Here's the nitrogenous bases: cytosine, uracil, thymine, adenine, and guanine. Anyway, but they are kind of important, of course, because of the storage of the genetic code that it does. Deoxyribonucleic acid is a double strand, of course. Ribonucleic acid is a single strand. So here's our ribonucleic acid right there, single strand, and here's our deoxyribonucleic acid, which is a double strand. And then ATP. ATP is pretty interesting. It's a nucleic acid, but it's not storing any genetic material. Its purpose is to store energy. And it stores a lot of energy. There are actually 2 bonds. So here is our model of ATP, right? We've got our adenine, we've got our ribose, and we've got our triphosphate. So adenosine triphosphate. Okay, that's important to remember. That triphosphate, that tri part, meaning that we've got 1 phosphate, 2 phosphate, 3 phosphates bonded together with these two energy-rich bonds. So to break that bond is an exothermic reaction. When that bond is broken, all of that energy in the bond is released and can be used in other ways. To make more bonds, to run certain machinery in the body. We're going to talk a lot about ATP because ATP is the energy molecule that your body uses to pretty much run everything. Then we get to proteins. And I will talk about proteins in a minute. Oh, I've only got 15 more slides. We're just gonna bust through. Proteins are extremely important. They're one of the most common of the macromolecules in your body, and they function in so many different ways. I'm starting to lose my voice. Catalysts, defense, transport, support, movement, regulation, storage. I mean, all of these things. Proteins are really important to the body. Now you do have a general structure, which is a chain. We're gonna have a chain of what we call amino acids. And these amino acids are going to be monomers, and you're going to have, 20 different types of amino acids, but depending on the arrangement of the amino acids in the full chain is going to determine what type of protein we have. So, yeah, these amino acids are covalently linked by a peptide bond formed by a dehydration reaction and split apart by hydrolysis. So let's take a look at a picture here. So here's our amino acid right there. We've got the amine, we've got the acid. And then right here, depending on what kind of an R group you've got, and when we say R group, we just mean it could be any number of different arrangements of molecules, but they're kind of arranged and then they're stuck on to the amine and the carboxylic acid. That R group could change. It could be an R group for a tryptophan. It could be an R group for arginine. It could be, I should know my amino acids, but I don't. But depending on what type of amino acid you've got, you're going to have a slightly different R group. And then as we put these 2 amino acids together, so here we've got one, here we've got another one, we're going to do a dehydration reaction and form a peptide bond. And as we form several peptide bonds between all of those different amino acids, we're going to see the protein chain form. So this right here is a monomer. This right here is a polymer. Single molecule, compound molecule, right? So the polymer is going to be a large molecule made up of smaller subunits, like the amino acid. So really, when you guys hear the word amino acid, you should think protein. When you hear the word peptide bond, you should think protein. When you hear the word protein, you should think amino acid and peptide bond. You need to have these words associate one with another. So the general structure of a protein is going to be, well, you've got these three, an polypeptide or protein. An oligopeptide is going to be between 3 and 20 amino acids, polypeptide between 21 and 199, and then a protein itself, a full protein, will be more than 200. A glycoprotein is something kind of weird. That's going to be a protein with a carbohydrate attached. Glyco means, sweet, I think in Latin or Greek, and then protein. So, it's this protein with a glucose on it. And so we organize them into groups based on their R group. Well, the amino acids we do. Amino acids, non-polar ones, contain R groups with hydrogens or hydrocarbons. Polar amino acids are going to have some other element. The polar ones can form interactions with water, where the non-polar ones cannot. Then we get to charged amino acids with special functions. I don't know. It doesn't really matter necessarily. You just really need to understand that an amino acid is going to be that subunit of the protein. So here's a few amino acids for your perusal. If they're in your book, I don't need you to memorize them, so you don't have to worry too much about it. But we do need to understand what a conformation is. The conformation is going to be the three-dimensional shape of the protein, and it is crucial. That shape, that specific shape of a specific protein is going to be crucial for the way the protein actually functions within the body. And that conformation, that three-dimensional structure, is going to be dependent on the arrangements between the amino acids. Let's talk a little bit about this, all right? Let's start with, I'm just going to skip that because again, we're looking at hydrophobic stuff. So let me explain. So right here, we've got a primary structure, okay? The primary structure of a protein is just going to be the linear chain of amino acids, all right? It's just the sequence of amino acids. Every protein has a primary structure that is the chain of amino acids. That chain of amino acids then is, we're going to see each one of these amino acids interact with one another. Notice over here that that these guys are kind of they're sort of bunching up a little bit, and it's because this amino acid is going to be attracted to that amino acid, and so we start to form this sort of structure beyond just the line of amino acids, because they start to stick to each other. And when they start sticking to each other, they're either going to stick in a secondary or a alpha helix right here, or a beta pleated sheet. And this is going to be our secondary structures, right? And when we hit this particular level, we call it the secondary structure of the protein. Now, what you need to understand too, though, is that this little molecule over here, he might start to interact with this molecule over here and start to like bend over to stick, right? Once we start to see all of that going on, we get to the tertiary structure. Tertiary structure is going to be a three-dimensional globular shape of the polypeptide. So again, we've got this fibrous protein, which is just one long alpha helix, but it's still interacting with each other, right? And then over here at globular protein, we're going to have this alpha helix interacting and sticking to this beta pleated sheet, which is interacting and sticking to this alpha helix, so on and so forth. Usually in class, I do this. I do a little exercise where I take a piece of paper, right? So right here, I've got my piece of paper, and this would be considered primary protein structure. Just a line, right? Just a line of stuff. Now, if I take this and I make it so that it's sticking to itself, or possibly twisting up on itself, at this point, now I have secondary structure. Okay, I've got a beta pleated sheet here. You can see the folds, and I've got an alpha helix here, right? Then I'm going to take that, I'm going to start sticking all of those different structures, the helices and the beta pleated sheet, together, and I'm going to kind of make sure that they're nice and tightly interactive. And now what I have is this three-dimensional structure that is a globular protein. I hope that makes sense. That's kind of how I do it in my face-to-face class. But tertiary structure, right here. My tertiary structure, globular protein. Now, quaternary structure is a little bit different. Quaternary structure is where we get, two or more tertiary structures and we stick them together. So hemoglobin is going to have 4 polypeptide chains stuck together. And then, we get to some of these other things. Prosthetic groups are non-protein structures covalently bonded to proteins. Like, a glycoprotein, you're going to have the protein and you're going to have the carbohydrate that's stuck to it, or the heme group, the lipid heme group and the hemoglobin protein. And then denaturation. Denaturation is going to be any conformational change to a protein. So if I take this protein and I take it and I change the shape a little bit, all of a sudden I've denatured that protein. It disturbs the protein activity. If I change the shape, all of a sudden it can't work the way that it's supposed to work. You can't see it too well. All of a sudden, the changed shape screws it up. Usually it's irreversible. It's not always irreversible. And it can occur due to 3 things. Increased temperature, right? So if we heat something up, it's going to denature those proteins, which is what happens when you take an egg. You take an egg and you've got that nice sort of liquidy protein full of a lot of water. You cook it and it changes from that gel to a nice rubbery solid. That is the denaturation. It's a conformational change in the protein. Or changes in pH even. For example, I don't know how many of you guys enjoy eating ceviche. I don't know if I'm pronouncing ceviche. Anyway, ceviche is, some of you know it, you take seafood and you let it sit in an acid. And the acid sort of cooks the seafood. So I mean, that's true. It kind of does. It's doing the same thing that heat is doing, except we're using a chemical process to denature the proteins. Word to the wise though, cooking seafood kills parasites because we're heating it up and killing anything still living in it. Ceviche is acidic, and so it doesn't actually cook it. It's not bringing it up to a certain temperature. So you want to be careful what kind of ceviche you're eating. You want to make sure that it's clean and free of parasites, because the chemical process doesn't always kill a parasite. And yep, amino acid sequence and protein conformation. There's more denaturation. That's it. This is it. We're done. I'm tired. This is a long presentation. So you guys, you're good. You're doing good. Keep going. Keep studying. I'm gonna get some water. Man, I'm looking haggard today. Anyway, and I'll see you when you on this recording later.